Rahim. Now we will start topic-wise MCQs, and I hope these are going to be helpful for you because then, as you finish a chapter, you can easily do the MCQs on that chapter. You can see in these questions, I mean, if you look at question number one, which organelles are enclosed in a single phospholipid bilayer? Now, you know it's only lysosome. Mitochondria has a double membrane. Golgi body has a single membrane. Endocytotic vesicles has a single membrane. But the fact was the hydrolytic enzymes was the word which was a key word, and that is not present in either the endocytotic vesicle or the Golgi body. Then the DNA of prokaryotes is naked and circular. Now here, of course, they're testing your English more than your biology because it says how is the DNA of prokaryotes. Now, like for instance, I say, okay, person X and person Y, what is the difference? And you say one wears glasses and one does not wear glasses. But you say, oh, one is taller. Well, we're not talking of the tallerness. So the DNA of prokaryotes, now here it's nuclear envelope, so this is wrong. Nuclear envelope, this is wrong. So the first two are wrong because they're talking of the nuclear envelope. But it says how the DNA is naked. Which statement describes how the DNA of eukaryotes? Now this is where you don't read this. It differs from prokaryotes. So the answer was D because it has proteins attached to it and is linear. Please remember both eukaryote and prokaryote are double helix. So double helix C was wrong because both are double helix. Then coming to the question number 3, the recently discovered Pandora virus measures 1000 nanometers, the Mary virus a diameter of 400 nanometers. What can be detected using a light microscope with a maximum resolution of 0.25 micrometer? Now immediately convert that into nanometer. Now you can see why the answer was A, because the resolution it means is what can be detected using a light microscope with a maximum resolution of 0.25 micrometer. This 2.25 micrometer is equal to 250 nanometers. So anything bigger than 250 nanometers will be visible. Anything smaller than 250 will not be visible. So that is why the answer was A, both the Miri virus and the Pandora virus. Mimi virus and the Pandora virus, because one was 400 and one was 1000. So both were more than 250. Please understand this. What do you mean by resolution? What can be detected using a light microscope with a maximum resolution of 0.25? Anything above 0.25 micrometer. So they now were testing whether you knew the units, whether you know how to convert centimeter to millimeter, millimeter to micro, and micro to nanometers. Question 4 and 5. Now 4, the answer was A. And that's very easy because wherever there is DNA and wherever there are ribosomes, so there will be mRNA as well. Of course, you might not understand it at this point, but when you've done chapter 6, then you will know this. Question 5. Until recently, the typical virus is 20 to 150 nanometer. Always underline it when you're doing this MCQs. In 2003, Mimi virus was discovered, which is 680 nanometer. In 2013, the Pandora virus is 1000. So you just have to write these here and then you would be able to figure it out very easily which one could be seen and which one could not be seen. And this is 20 to 150. So 150. Now it says which virus can be seen both a light and a maximum resolution of 0.25 micrometer electron microscope. So basically anything which was above 250 nanometer would be seen. So that is why the answer was B. In question 6, you had to measure this XY, measure it on the paper, whatever the length, convert it into micrometer, and then divide it by 210. And you would have got the answer. Then coming on to question number 7. Now, this is the one which is more difficult, and you're all. No, 7 is not difficult, 8 is difficult. Okay. In 7, what is the correct order in which organelles function to make and secrete an enzymes? You might not know it at this point, but it's nucleus, RER, Golgi, and vesicle. You will understand it better when you've done chapter 6. Then let's come on to question number 8. These are the ones which really haunt and daunt you. So let's go through it very carefully and very slowly. And I hope this is going to be clear to you now. Now I want you to pause this video here for a minute and look at the working and then listen to what I have to do the explaining. The diagram shows a stage micrometer scale with divisions 0.1 millimeter apart. So these dark lines is the stage micrometer, and that is 0.1 millimeter apart, viewed through an eyepiece containing graticule. So this is the, if I just shade this in yellow, this is the eyepiece graticule, which we call EPG, eyepiece graticule. So this is the eyepiece graticule, which I am shading in yellow, which I've just rubbed off. So this is the eyepiece graticule, and this is the stage micrometer. Always this is the rule. So now you can see how I have done the maths for this. So 0.1 is equal to 40. From here to here it is 40. So 40 is equal to 0.1. Now the millimeter you have to convert into micrometer. So 40 is equal to 0.1 into 1000 micrometer. And then 40 is equal to 100 micrometer. Then 1 is equal to 100 over 40 into 100. And 100, why 100? Because this whole thing is 100. This is the diameter of the field of view. This is the diameter of the field of view of the microscope. So 100 250 hai. Now the formula is pi r square. So pi is whatever is given to you, 3.14. R is 125 r square. R square means r into r. This would be equal to, you see the working, which is correct, and that was the answer. So please understand this. Please pause this video here, have a look at it again, and I will just remove all the markings so that you can all see it and then fix it up. So please take a screenshot of this here, and then I will remove all the, and now you can do it on your own and check whether you've understood this or not. This is the one of the questions which a student asked me today, and I said, give me the number of the question that you don't understand, and she said she didn't understand 9, and I said to her, this is a question which checks whether you know the sizes of the organelles. A scientist carried out an experiment to separate organelles in an animal cell by mass. The scientist mixed the cells with a buffer solution which had the same water potential, because you don't want the organelles to burst. He then broke the cells open in a blender to release the organelles. The extracted mixture was filtered and then spun in a centrifuge. Centrifuge means you spin it so fast that the heavier ones will settle at the bottom, and then of course the lighter one will go on the top. To separate the heaviest organelle, this sank to the bottom, forming a solid pellet 1. So the heaviest would be in 1. The liquid above the pellet, so this liquid above the pellet, which contained the lighter organelles, was poured into a clean centrifuge tube and spun in the centrifuge at a higher speed to separate the next heaviest organelle. All this story is in the question, but if you bother to read the question and understand the question, just don't get angry and agitated. This organelle sank to the bottom, forming a solid pellet 2. So the biggest organelles, then the one smaller than that, and then the one even smaller than that. He repeated this procedure twice more to obtain pellet 3 and 4. So, I mean, I haven't made this very correct, so I'll just correct it. So the fourth one was the smallest. So this is pellet 1, pellet 2, pellet 3, and pellet 4. What is the function of the organelle extracted in pellet 2? Now, if you have understood this chapter very well, digestion of old organelles, what is that? That is lysosomes, right? Production of ATP, mitochondria, right? Production of mRNA, nucleolus, sorry, nucleus, not nucleolus, that's production of ribosomes. And synthesis of proteins is what? It is ribosomes. Now pause the video here and look at it very carefully. Digestion of old organelles, lysosome, production of ATP, mitochondria, production of mRNA, nucleus, synthesis of proteins. So, which one is the biggest organelle? The biggest organelle is nucleus. Then this is 1. Then this is 2. Then this is 3. And then this is 4. So it said, what was pellet 2? So pellet 2 had to be mitochondria. I mean, we've done this in class also. the different sizes of the different organelles. And I've done this whole exercise with you. Question 10, which is the correct comparison, typical prokaryote and typical eukaryote? Typical. Now, why is this wrong? Because eukaryotes also have circular DNA in their mitochondria and chloroplasts. And most of one would have thought of A. I know this because you were not reading it correctly. So eukaryotes have linear DNA, I agree with that, but of course they have circular DNA in their mitochondria and chloroplast, but you wouldn't have thought of that. Of course, this is all beautiful story, BS. There's no rough endoplasmic reticulum in prokaryotes, and you know that the prokaryotes diameter is smaller than the eukaryotic diameter. Again, reading disorder. Question 11, a student was presented with a photomicrograph of a cell organelle. The magnification of the photomicrograph is shown. So there is something written here, X something, which is the magnification. Now, which calculation of the actual length of the organelle in micrometers is correct? Now, you didn't read this micrometer. So the image size in millimeters, say this was 10 millimeter. So you multiply it by 1000 to get micrometer. And you then divide it by the magnification, whatever was given here. So the answer was D. Then question 12, which statements both about mitochondria and chloroplasts are correct? This is wrong. ATS नहीं होता mitochondria and chloroplast में. There is no ATS ribosome in mitochondria and chloroplast. Yes, they contain circular DNA and they produce ATP. This is a point of the syllabus. Produce ATP, both of them. So the answer was 2 and 3. Which range of sizes would include most eukaryotic cells? And then you of course ask me, Miss, do we have to know the sizes? Yes, you have to know the sizes. Roughly, you've got to know the range of sizes. Then we come to question 14, which is very much like question 9. But here they have asked now pellet 4. It's the same question. But now they have asked pellet 4. So this was lysosomes, this was mitochondria, this was nucleus, and this was ribosome. So now they have made it into pellet 4. So that is pellet 4 would be ribosome, would be the last one. Go back to question number 9 and have a look at it again. Question 15 and 16 are very basic. Which size of ribosome found in mitochondria? 70s. Different units are used with measuring biological specimens. Which measurement in millimeter has not been correctly converted into both micro and nanometer? So I'm sure everybody can figure that out. Then which features of microvilli and root hairs are correct? Increase the cell surface area. Cannot be resolved with a light microscope. Contain vacuoles. More than one present on a cell. So the answer was C. Then lysosomes have a variety of different shapes, making them difficult to identify. What describes a lysosome? Basically, it was this. This was wrong here. Double membrane nahi hota. Lysosome doesn't have a double membrane. It is a single membrane lysosome. Mitochondria have a double membrane. Chloroplasts have a double membrane. Nucleus has a double membrane. So this is how you pick up what is wrong and what is correct. So this double membrane was wrong. So this was wrong here. I would have cut it out immediately, which is the wrong ones. And then I would have sort of thought about B and C, which one is correct. So both of them have a single membrane. But why is it wrong? Because B was a vesicle containing hydrolytic enzyme surrounded by a single membrane found only in phagocytes. No, that's rubbish. They're not found only in phagocytes. They're found in other this thing also. Question 19, radioactive labeled amino acids are introduced into a tracheal cell that uses them to make mucus a glycoprotein. You know glycoprotein would be Golgi. So first of all, the amino acids would be present in the cytoplasm, so it's 1. Then the protein would be synthesized on the ribosome, which is 4. And then it would go to the Golgi, which is 3, because it will add the glycogen part to it. And then it will form a secretory vesicle, and then it will go out of the cell in exocytosis. So what route will the amino acids take? This is for those students taking the October-November exam. They can revise from here as well. Which structures are present in a Vibrio cholera cell? Now, of course, they're now asking you diseases has been done as well if this is the case in which a student taking the AS. So bacteria. which presents are present in a Vibrio cholera. Vibrio cholera is a bacteria. So it has cell wall and circular DNA, but it does not have ATS ribosome. That's why the answer was D. What is the diameter of a typical plant cell? I mean, this is what you have to figure it out, and you need to know this very well. Question 22 checks a lot of very important skills. A specimen is viewed under a microscope using green light with a wavelength of 510. Why have I divided by 2? Because you can see anything less than half the wavelength cannot be seen. So the resolution will be 255. 510 is the wavelength, crest to crest. This is the wavelength. So half of the wavelength, if anything is less than half, then you can't see it. It has to be bigger than half so that then it interferes with the light. And that the light is reflected, and that's how you see things. Now, if the same specimen is viewed under the same conditions, but using red light with a wavelength of 650 nanometers, so now it will be 650 over 2, that is 325. So first you could see dots, which were, you can see these as two separate points when they were 255 nanometers apart. Now you will see the dots which are 325 nanometers apart. So now what has happened to the magnification? Magnification is the same because it says viewed under the same conditions. So magnification is the same. This is why I underlined that. But now you've used red light and the wavelength is 325. So the resolution has decreased from the 2 points that were 255 nanometers apart. Now you have to see things which are 325 nanometers apart. So the resolution has decreased. Now you cannot see that detail. Don't use the word cannot see it clearly. Now you cannot see that detail. What is the function of plasmodesmata in plant cells? Question 23. To allow the symplastic movement of substances between cells. We haven't done symplast up till now, but still some of you might have done it. Which size of ribosome is found in chloroplast and mitochondria? 70S, very basic question. Which function is correct for the description of the cell structure? For the description of the cell structure, organize microtubules. Now that is the centriole. Package hydrolytic enzymes used in cell. Package hydrolytic enzymes. So the Golgi body does that. Synthesize lipids, SCR. Synthesize polypeptides, RER or ribosomes. So you can see which one was the matching part. Now the next question, 26. 26, ribosome consists of two subunits, each containing ribosomal RNA. An analysis of all the 70S ribosomes from a single cell of the bacterium Escherichia coli showed that they were 38,000 rRNA molecules. But it says ribosome consists of two subunits. So this is one subunit and this is the second subunit. And each one is made up of ribosomal RNA. So they were 38,000. So there must be two main types. So 19,000 one type and 19,000 two type. And there were 19,000 copies of each type of rRNA molecule. How many 70S ribosomes were there? Naturally, 19,000 because the ribosome was made up of two subunits. So 19,000 one type and 19,000 two type. Read the question again, understand it, and then reread it again, and then see why am I saying this. Combination of lenses for a light microscope will give the greatest magnification. You just have to multiply the eyepiece with the objective. 5 into 100, 500, 10 into 40, 400, 15 into 40, 600, 15 into 100, 1500. So naturally D was the highest. Which steps are needed to find the actual width of a xylem vessel viewed in transverse section using a 10 objective lens? Convert from millimeter to micrometer multiplying by 10 raised to the power -3. Calibrate the eyepiece using a stage micrometer. Measure the width of the xylem vessel using an eyepiece graticule. Multiply the number of eyepiece by the calibration of the eyepiece. So the answer was only 3 and 4. The statements are all description of cell structures. Surrounded by a single membrane enclosing a large fluid-filled space, surrounded by a single membrane enclosing inactivated enzymes, formed by two membranes enclosing a matrix, mitochondria, formed by a membrane that is flattened sacs and tubular structures interconnected throughout the cell, Formed of nucleic acid and protein attached to membranes or free in the cytoplasm, ribosomes. Which row shows the typical cell in which these cell structures are found? Now, 1, 3, 4, and 5 are found in plant cells. So 1, 3, mitochondria, 4, and 5, ribosomes, nucleic acid, RNA, and protein. You haven't done this as yet, but of course you will do it later on. So if somebody is still on chapter 1, then it will be difficult for them. And I can understand. You'll have to come back to it after chapter 6. And 1, 4, and 5 surrounded by a single membrane enclosing a large fluid-filled space. So the answer, as you can see, is this. The 1 is the large vacuole, which is only found in plants. So then you have to only figure out between A and B. 2, 3, 4, and 5 are the only ones which are present in animal cells. So 2 is single membrane, inactivated enzymes, lysosome. 3 is mitochondria. 4 is flattened sacs, RER and SCR. And 5 is the ribosomes. Question 30, I know you can just pause it here. We've done it before. 31 is also, we've done it before. Ribosome, RER, vesicle, Golgi. And at approximately which magnification is light microscopy not suitable because the resolution becomes too low? I've told you this in class as well. It's X 1500. After that, that's the maximum useful magnification. After that, it's useless. Now, question 33, which you all find difficult. A number of students have asked me this question. I'm going to just explain it to you. 33, the diagram shows a stage micrometer viewed with the eyepiece reticle scale using a magnification of X 400. Using the same magnification, a chloroplast is measured and found to be 4 eyepiece graticule. How long is the chloroplast? Basically, the magnification was just to make you confuse you. That was just not needed. You just have to say is 40 is equal to 0.1. This is the 0.1. This is the stage micrometer. So 1 will be 0.1 over 40 into 1000. Why 1000? Because we are converting it into micrometer. Because all the answers are in micrometers. If you look at all the answers, they are in micrometer. So 40 is equal to 0.1 millimeter, 1 is equal to 0.1 over 40 into 1000. Why have I done 1000? Because I want to convert it into micrometer. Or you could have done the final answer also into 1000, it's fine. So 0.1 over 40 into 1000, and then because we had to calculate 4, because it said 4 eyepiece graticule units. So that is the answer was 1 into 1.0 into 10 raised to the power 1 micrometer. Which organelle does not contain a partially permeable membrane? Yeah, Golgi, lysosome, mitochondria all have membranes, but the ribosome does not have a membrane. So this is what you have to know. It's made up of rRNA and protein. The diagram shows an electron micrograph. I've told you to look on a Google search and look at different micrographs. These are very favorite questions of ours. Now, of course, you can see that this whole huge thingy is the nucleus, the biggest organelle. And this inside it would be the nucleolus. And these, of course, are the chloroplasts, because something smaller than the nucleus is the chloroplast. And of course, you can see these. I'm going to use another color. These are the mitochondria, which you can see here. You can see here because they've cut in different sections, and you can see here all these mitochondria. And this, of course, is the large, the tonoplast and the cell sap, which is, of course, this is X is the cell sap. So diagram shows an electron micrograph of a plant cell. What do structures X, Y, and Z contain? So X is, of course, the cell sap, so it contains mineral ions. That's why cell sap was wrong. It says contains. And Y is, of course, the chloroplast, so it contains starch granules. And Z is the nucleolus, which contains DNA and RNA, because nucleolus is the site of ribosome synthesis, and ribosomal RNA are present in the nucleolus, where from the DNA, the ribosomal RNA is going to be made. Technically, a very difficult question. 36. An amino acid enters the cell, is then used to synthesize an enzyme secreted by the cell. So first it will have to be made on the ribosome, then the ER, then the Golgi, and then the exocytotic vesicle. The diagram shows some similarities between chloroplast, mitochondria, and typical prokaryotes. So you see, when you see this sort of Venn diagram, they're comparing them. So you have to find out which row is correct. You know, 70S ribosome is present in both chloroplast and prokaryotes. Also, 70S is present in prokaryotes and mitochondria. So this is the way you have to figure it out. All the ATS were all wrong because there is no ATS in any of these. There's chloroplast में there's no ATS, mitochondria में नहीं है, typical prokaryote में नहीं है. 38, which structures measured in the unit shown using a light microscope? Cell surface membrane is in nanometers. Which structure is measured in the unit showing using a light microscope? But you can't see a cell membrane in the light microscope. It's only 5 to 10 nanometers. Cell wall is very, very thick. It's 5 micrometer. Chloroplast is micrometer. Ribosome is in nanometers. So that's why the answer was C. Then 39. A student studied two photographs, same magnification, palisade, one using a light, other electron. The student observed which features can only be seen because of the higher resolution of the electron microscope. I keep on telling you, you see more detail. If you see more detail, well, you see more detail is you see the structures, which are, of course, very, very detailed. And 1, 2, and 3. 1 is the cisternae, and 2 is the grana, and 3 is the two membranes of the nuclear envelope. Those are the very finer details, and of course, which are only in nanometers. That's why you can only see them using the higher. Of course, the vacuole enclosed by the tonoplast is a big structure inside the plant cell, so that's a huge structure. A piece of mammalian tissue was homogenized and centrifuged. Homogenized means it's put into a blender and it's made into a uniform solution. Look up the word homogenized in the dictionary. The biochemical activity of 4 subcellular fractions was investigated. Which diagram indicates a fraction with a maximum synthesis of messenger RNA? Now, you know the messenger RNA is made inside the nucleus. So which one has a big nucleus? The bar should be more of the nucleus. Now, this is the smallest. This is the smallest. So the highest was that. Now, I'll change that question and I'll say which one has the most hydrolytic enzymes. Then you go to look at the lysosome, and the lysosome bar is the highest. Let me give it another color. The highest bar is here. I know this question has come again. Lysosome, which will have more hydrolytic enzymes. So then the answer would have been C. So it's very easy. You see, you have to know the functions. You have to know the functions of each organelle, and then of course you can do any MCQ. It's not very tough. I mean, it's you have to just learn these things. Which row shows features of a typical eukaryotic cell? I'm sure everybody understands that. Then it says many singled animals live in fresh water, possess vacuoles, which contract regularly, expelling excess water. And why do the cells of plants living in fresh water not require such vacuoles? Plant cell walls limit cell size. I've never understood why this was the answer, but I'm sure you can figure it out. I sometimes some of the MCQs I find they are too confusing for me. I think we should end this here and I will continue. There are another 40, I think, MCQs on this chapter, and I will continue it and hopefully upload it tonight. And I hope this has been helpful. Please do subscribe to the channel and do write your comments so that I can make it a more helpful channel for all the A-level students taking their biology exams. And thank you very much.